Protéus did a very interesting talk yesterday about some application on lipase discovery, and I'm pretty sure that Ronan will have some discussion about this. Thank you, Ronan.
So thank you, Egon, for your introduction. I will share my screen. So for those who already assisted to the presentation of my colleague Olivier Vidalin, who is a scientific manager at Protéus by Seqens, there will be some common parts. I'm Ronan Rocle. I'm a business development manager for R&D services within the group Seqens. And I will present you today first the group Seqens, its activities, our subsidiary Protéus, which is specialized in biocatalytic processes development and directed evolution technologies to improve the performances of enzymes. And I will present you some examples. So let me put it on full screen mode. Okay, so first, let me present the group Seqens. Seqens is a specialty chemicals company developing pharmaceutical ingredients as well as fine chemicals for its customers. So we are a company of more than 3,000 employees, headquartered in Lyon, in France. We have 24 manufacturing sites all over the world and 3 R&D centers. The main one is located close to Paris, in Porcheville. It's the main R&D center for our chemical synthesis. A second one is located close to Boston for pharmaceutical applications. And the third one is actually Protéus, so Protéus by Seqens, which is located in the south of France, in Nîmes. So actually about the activities of the group Seqens, we are, let's say we have 2 main activities. The first one is on pharmaceutical solutions. So we are both a large volume producer of, let's say, commodity, let's say large volume pharmaceutical active ingredients, such as aspirin. We are the leading producer worldwide of aspirin, and we are also a very large producer of paracetamol. So that's for the active principles that everybody knows. But we are also synthesizing the complex molecules that are developed either by big pharma or by biotech. So for these companies, we offer our services first at R&D scale to develop an efficient synthesis using multiple chemistries to efficiently synthesize the active ingredients they have discovered. And then we can accompany them using our flexible manufacturing sites in order to produce these active pharmaceutical ingredients for the clinical phases up to commercialization. So that's for one side of the activities of Seqens. Also, following COVID, actually we started another activity which was for actually disinfectant solutions. For example, that's hydroalcoholic gels actually, and also solutions on sanitizers, which were widely used and are now one of the an additional activity of the group Seqens in terms of pharmaceutical solutions. And we have also another activity which accounts for 30% of our results, which is actually the non-pharma sales on fine chemistry. So it can be, for example, in the field of cosmetics. We are the leading producer of salicylic acid. We are also a large producer of cosmetic solvents such as isopropanol, for example, and we can make custom chemistries for the synthesis of the active ingredients for cosmetics, as well as polymers in electronics, which require a very high purity in terms of the impurity in terms of metal has to be below parts per billion. And we have actually a large diversified activity in the fine chemical space. So that's for the overview of the group Seqens. And among this group, as I explained, we have the subsidiary Protéus, which is, let's say, the biotechnology center of the group Seqens. So let me present you briefly the activities of Protéus. So Protéus was created more than 20 years ago, initially as an independent company, and it was progressively incorporated within the group Seqens. With a joint venture that was created between PICAS, one of the members of the group Seqens, and Protéus in 2006. It was in order to find synergies. Protéus was helping PICAS to develop biocatalytic processes as a way to more efficiently synthesize active pharmaceutical ingredients as well as specialty chemicals. And Protéus is now fully part of the group Seqens. So the expertise of the group of Protéus is in the development of tailored enzymes and also biocatalytic processes development. So we have a team of chemists specialized in biocatalysis. We have also a team specialized in molecular biology in order to improve the enzymes using technologies such as directed evolution. We have also a team specialized in microbiology. So we have a very large and diversified collection of strains, and a large share of them have been sequenced. And that's a mine that we use as a start for our projects in order to find new enzymes with improved activities. We have also a team specialized in fermentation, which allows to perform the first steps of upscaling of biocatalytic processes. So that's a multidisciplinary team of 25 employees, which has all the capabilities to, let's say, to lead a project for our customers, starting from a need, a specific chemical reaction, to identify a new enzyme which would be able to catalyze the target reaction, to improve this enzyme, starting from exclusive libraries of sequences, using also technologies such as protein engineering, and then here we are supported by the group, and in particular by the R&D teams close to Paris. We can upscale the processes up to ton scale. So using the pilot scales of the group Seqens as well as commercial scale manufacturing. So that's, let's say, a full service that we can offer to our customers for the development of enzymatic processes. So here we present actually the main capabilities of Protéus by Seqens. The first one is that during the 20 years of existence of Protéus, we gathered a very large collection of strains coming from, in particular, from extreme environments. So the goal of this collection, in fact, is to identify enzymes which would resist to the harsh conditions you can find in chemical process. For example, high temperature or low, also acidity, salinity, or pressure. So using this collection, and we have more than 1,000 enzymatic activities actually that have been, which have been developed, let's say, which have been fully sequenced. So we have access to the genomes. We can look into these exclusive collections to find new enzymes which are very relevant and with higher performance for our customer reactions. So that's a way. Also, it helps to find, let's say, naturalness. For example, in the cosmetic sector, we can identify native enzymes which are very active on such reactions. The second aspect is protein engineering. So we are able to develop tailor-made enzymes, also to modify actually the amino acid sequence of enzymes in order to improve their activity. So for that, we have two directed evolution technologies which are proprietary to Protéus, and we successfully used in several customer projects. And the last part is the ability to upscale processes. So we are integrated within the group Seqens, and we are able to upscale reactions from, let's say, the proof of concept to the ton scale. So actually, as an example of this, we have developed, for example, an aldolase process in order to make the non-natural amino acid D-serine. So in this example, in fact, the enzymatic synthesis, enzymatic pathway is clearly superior to what you would do actually with a standard chemistry process. You don't need protecting group. It's a very selective synthesis that is operating at ambient conditions and with a very high enantioselectivity. So we just have the D serine, not the L serine at the end of the process. So in this case, we are producing actually the enzyme and we are using it directly for the reaction. And at the end, the enzyme residues are filtrated and we get a D-serine, which is at the right quality to be used for the synthesis of active pharmaceutical ingredients. And we calculated actually what was the cost of the enzyme in the process, and the impact of the enzyme in the production cost is limited to 6%. So that's actually a very small part of the cost for a product which is produced at the scale of dozens of tons per year in our site of Couterne in Normandy. And that's something actually that we could generalize. So usually for the processes that we developed for our customers, the cost of the enzyme for the conversion, the total conversion process is less than 10% of the total cost. So, I mean, it's not the enzyme which is heavily impacting the cost. The enzyme as a catalyst is a very efficient catalyst, either at smaller scale for pharma, but it's also true for food or even for commodity chemicals. We have also other examples. So, for example, this is the way we are using the diversity of our collections in order to source enzymes of interest. So we have a lot of extremophiles, and we use that to identify enzyme with a particular and superior performances in specific environments. So I won't detail on this example, but all our collection has been gathered in respect of the Nagoya Protocol. It's not a metagenomic collection, so there is a one-to-one relation between the genomes and the conditions where the microbes grows and can actually, when you can get some efficiency of the enzyme. So it's a very clear relation between them. And we use our collection to source new enzymatic activities, which can prove superior, for example, to standard activities. So here it's another example. It's a confidential project that we laid for a pharma customer for the synthesis of a chiral API at ton scale. So in this case, this is another example actually where we developed a tailor-made enzyme, and we found that its performances were clearly superior to what you can find in commercial enzymes. So you can think here, we found an enzyme that is active at a very low concentration, 400 ppm, which gives actually a very high enantiospecificity, and we just had to produce it in 40-liter fermenters in order to be able to synthesize this chiral intermediate at ton scale. So this was actually a very good success, which shows actually the superiority of tailor-made enzyme when it's optimized for the synthesis of many specialty pharmaceutical ingredients or specialty chemicals. And in this case, the enzyme cost was only limited to 7% of the total cost. So I finished my presentation, and you can see Protéus in our booth, or you can directly email me if you have any questions about the presentation. Thank you very much for your attention.